Okay, let's continue our topic of carbohydrates. Now we will be discussing about polysaccharides. Carbohydrates composed of 3 to 10 monosaccharides units are classified as oligosaccharides. Carbohydrates with higher than 10 monosaccharide units are classified as polysaccharides. Despite the availability of reactive anomeric carbon in some oligo or polysaccharides, the long carbon chain decreases the polarity of the whole structure and diminishes its reactivity. That is why all oligo and polysaccharides have no reducing capacity. Based on the type of monosaccharides unit, polysaccharides are also called with a specific name. For example, the polysaccharides made with glucose is called glucans. We'll discuss about several examples of glucans in this lecture. While the polysaccharides made with fructose is called fructans, those from mannose is called mannans, and those from xylose is called xylans. Another term for the polysaccharides is the glycans. So you have glucans and you have glycans. Glycans is another term for polysaccharides. Glucans is another term for polysaccharides made from glucose. Based on the uniformity of the monosaccharides unit, polysaccharides can be divided into homoglycan and heteroglycan. Homoglycan are polysaccharides that are made up of single monosaccharides. For example, starch, cellulose, glycogen, and inulin. Heteroglycans are polysaccharides that are composed from 2 or more monosaccharides or their derivatives. For example, agar, gum, hemicellulose, pectin, hyaluronic acid. Homoglycan can be further classified based on its function. For storage, such as starch in plants, glycogen in animal, inulin in roots and rhizome, and dextrin in bacteria and yeast. And structural, such as cellulose and lignin in plants, and chitin in insects and crustaceans. Heteroglycans perform diverse functions. It can be structural, water metabolism regulation, cellular cement, biological sieve, biological lubricant, docking site for growth factors, and other functions. Since quite many of heteroglycans are formed by the repetition of the saccharide unit of an amino sugar and an acid sugar, some has classified heteroglycans into nitrogenous heteroglycans and non-nitrogenous heteroglycans. Okay, next.
Polysaccharide can also be classified into their polymerization pattern. The patterns include linear polymers such as amylose and cellulose, branch polymers such as amylopectin and glycogen, interrupted polymers such as pectin, block polymers such as alginate, and alternate repeat polymers such as agar and carrageenan. Polysaccharides are also referred as hydrocolloids or food gums. This is because it is often used in food to thicken and or gel aqueous system. But you see, some polysaccharides such as agar can create a gel that holds up water to more than 1,000 times of its own weight. Pectin in some fruits may create a jam consistency with an addition of acid. While if you add cornstarch to your hot soup, what you get is a consistency like a puree. Difference of each polysaccharide's ability to modify, control, and even to stop the flow properties and textures of liquid products are very much related to their structural properties, including their polymerization pattern. Okay, next.
Starch, cellulose, and glycogen are 3 most important glucans. They are all homoglycans with glucose for their monomer unit. As previously mentioned, starch is an example of storage glucans, while cellulose is an example of structural glucans. Their functions are highly related with the glycosidic linkage connecting their monomer. Though they are both polymer of glucose, starch is mostly connected through alpha-1,4 glycosidic linkage, while cellulose is connected through beta-1,4 glycosidic linkage. All plants contain alpha-glucosidase enzymes such as amylase, that able to break down the storage glucans of starch into simple sugar that are soluble in water and easily transported within the plants. This mechanism is very important in the plant germination and also in the fruit ripening process. That's why ripened fruits is sweet. While beta-glucosidase such as cellulase is only released as part of plant defense mechanism upon tissue disruption. So, while the starch is being hydrolyzed during the plant growth, cellulose remains intact. Upon cell disruption, beta-glucosidase is released to activate phytoanthocyanins. These are immediate chemicals that is a defense mechanism against protruding herbivores and pathogens. And the release resulted in the hydrolysis of cellulose as side effect. This might explain why if your apple is bruised, the texture around the bruise also got mushy. Okay, next. I previously mentioned that starch is mostly connected through alpha-1,4 glycosidic linkage. Well, there are two kinds of glucans in starch, amylose and amylopectin. Amylose is a linear chain of glucose all joined by alpha-1,4 glycosidic bond. While amylopectin too mainly joined by alpha-1,4 glycosidic bond, it also has some alpha-1,6 glycosidic linkage every 24 to 30 glucose units. This creates a branch structure. This branch structure causes amylopectin to be less compact compared to amylose, but also provide better availability for enzyme to hydrolyze the bond. That way, amylopectin can provide glucose faster in cellular respiration. Okay, next. Now, after talking about glucans in plants, let's talk about glycogen. Glycogen is a homoglycan from glucose units that function as glucose storage in animal. In a way, glycogen has a similar structure with amylopectin in terms of the existence of alpha-1,6 glycosidic linkage that form branching. However, glycogen alpha-1,6 linkage happens every 8 to 12 glucose units, creating higher degree of branching and more rapid hydrolysis in time when glucose fuel is needed. Okay, next. So, by now, we have taken a look into 4 examples of glucans, namely amylose, amylopectin, glycogen, and cellulose. They are all polysaccharides with glucose as their monomer. But the difference in glycosidic linkage that joined the glucose units resulted in different structures and functions. Let's review a little bit about 4 of them. Okay, get ready to answer my questions. Do you know which of these glucans are linear? Next, do you know which of these glucans are branch? Next, do you know which of these glucans work as carbohydrate storage? Next, do you know which of these glucans is a structured carbohydrate? Next, 2. Amylose and amylopectin are both starches. They have different in structure and also speed of glucose release. Can you explain that? Next, 2. Amylose and cellulose are both linear. They have different structure and function. Can you explain that? And last, amylopectin and glycogen look pretty similar, but glycogen have higher degree of branching as animalic storage of carbohydrate. Why do you think so? Okay, that's a wrap. So this is our review points for carbohydrate. After watching this video, can you answer these questions? Why do we need to learn about carbohydrate? What is carbohydrate? What is the carbohydrate classification based on degree of polymerization? What are the nomenclature of monosaccharides? What are reducing sugars? What are some important simple sugars in food biochemistry? What is glycosidic linkage? How to classify polysaccharides? And what are some important examples of glucans? Okay, That's all for today's review about chemical structure of carbohydrates. For questions and comment, you know where to contact me. See you on another lecture.